Okay, guys, so we are now going to finish this chapter, The Ways of Change. And this is the last chapter that will be included in the test number 2. So last time, we pretty much went through most of this chapter, most of this lecture. And remember that our scope really is to finally understand how allele frequencies behave in populations, which is a point that we have not discussed yet. But in order to understand this point, we needed to understand a little bit of genetics. We needed to understand what DNA is, where genes are located in our genome, and especially how what are the main sources of genetic variation in populations. All right, so we already discussed all these points, so we're now going to jump on allele frequencies and how they behave, how they're studied in populations. So I'm going to go fast to this slide. All right, so we're going to talk about allele frequencies. So remember the concept of natural selection. Natural selection, that the concept of natural selection, evolution by natural selection, that was introduced by Darwin, if you remember. And one of the main points upon which natural selection is based is genetic variation. There is an intrinsic variability in populations. We know now how this variability arises. We have studied that in the first part of this presentation. Therefore, if there wasn't genetic variation, there would be no natural selection or no evolution. So differences exist in DNA exist in individuals within a species. We're all different, and those differences are the base of evolution of a species, change over time. Now, what we're going to understand today is how allele frequencies behave in populations. Remember that whenever we talk about a gene variant, we don't talk about mutation. We talk just about allele, because we cannot say that a person that has blue eyes has a mutation compared to a person that has brown eyes. It won't be really fair. It's not a mutation. It's just an allele variant, a gene variant or allele. Okay, so in populations of a species, we can many times recognize several alleles that all contribute to certain phenotypes. In this example, for instance, you have here you're presented with an example of, for instance, this could be a species of plant that produces flowers of 4 different colors, maybe white, red, yellow, and purple. And these represent the 4 alleles or gene variants that are present in the population. And based on the combination of these alleles, remember that for all animals, we talk about diploidy. So you can only carry 2 of these allele variants at a time, not all together, because you only carry 2 chromosomes for each copy. But I have to say that in plants, sometimes you have the so-called polyploidy. It's just a curiosity, I'm telling you. I'm not going to ask you to describe this, but plants, some species of plants may carry more than 2 chromosomes for each representative. Anyways, let's consider, remember in this course we focus on animals specifically, so we always talk about diploidy, okay? And this example could be an example of also a gene human population, because remember I told you that talking about eye color, for instance, in human population, there have been at the very least 6 different alleles that contribute to eye color in human population, not just 2 or 3. Okay, and depending on the combination of these alleles that you carry in your genome, that determines the eye color. There have been at least I think 9 types of eye color recognized in human population. Okay, so many times we have more than 2, 4, 5, 6 different alleles that are around in a population of a certain species to regulate a certain genotype and so phenotype as a consequence. Okay, so as I said, this would be an example of flower color in a certain species of plant. Okay, and in this case, in this very example, it shows you that it wants to show you that the frequency or fraction of each of these allele in the population of this species is perhaps 25%. So statistically, we see that 25% of plants are this color, 25 this color, 25 this color, and 25 this color. That's one possibility, okay? And so, so in a population of, say, 1,000 flowers, an individual can be red, white, blue, or yellow. But remember that all 4 colors are present in the population in form of alleles, okay? So when we talk about allele frequency, frequency is a fraction or is a percentage. They're all the same thing. In other words, you can say, for instance, the allele for white flower is represented 1/4, that would be a fraction, or it is present 25%, which again is just a different way to express the fraction 1/4. All right, so or you can say 1/4 as well. So a percent is a type of proportion, okay, and you should be acquainted with this concept already from math, from school math. And so we can talk about, when we talk about frequencies, again, we can talk about proportions, percentages, fractions, are all the same thing. All right, so now, how do allele frequencies behave in populations? Let's say if you have a population that at this moment presents a certain fraction of certain alleles, how will this fraction behave over time, over the course of generations? Will it be always the same? Will it change? So these questions were asked especially by a mathematician. His name is Hardy Weinberg, and he studied these allele frequencies, and he actually was able to come up with mathematical models that describe how allele frequencies in populations behave. Okay, so Hardy Weinberg came up with an equilibrium model, which is known as the Hardy-Weinberg equilibrium model. And because in order for him to come up with some kind of mathematical formula, he needed to base his formula on a model. And then there could be deviations to this model. Excuse me. Otherwise, he could never come up with mathematical interpretation. All right. So now, in order to understand the Hardy-Weinberg equilibrium model, we have to understand what are the premises to this model. Remember, this is a model, so there are some premises to it. So, first of all, what does the equilibrium state conceptually? The equilibrium model states that allele frequencies stay the same or don't change from generations to generations over time if some conditions are met. Conditions or assumptions, we can call them. What are these assumptions? No mutations are occurring in the population. large population sites. This is very, very important point. And there is this link here where you can simulate the what happens actually to these equilibriums when you change population size. And what you can see is that the equilibrium is disrupted as the population size is reduced. So large population size is very much of a basic important point for the Hardy-Weinberg equilibrium model to be satisfied. No major mutations are occurring. So of course, if all of a sudden, for instance, in a population, there are strong factors that increase the mutation rate to higher than the very, very minimal. For instance, imagine a population where that is hit by strong radiation. All of a sudden, this equilibrium could be disrupted. No movement of individuals and their alleles, of course, in or out of the population. No migration out, no migration inside the population. As you can imagine, for instance, human populations of different places of the world, they carry different alleles because they have been subject to different environmental pressure, different natural selection path. And therefore, if you have that some populations all of a sudden come in large number in a pre-existing population in a certain place, this is going to disrupt or alter an equilibrium. Another important assumption is that random mating is happening in the population. Of course, if somehow artificial mating is happening, it's kind of like almost doing artificial selection, and this would definitely disrupt that randomness and equal possibility of allele combinations. Okay, so you don't want you want random mating to happen among individuals in the same population. And therefore, with all that said, you don't want natural selection events in course, because if natural selection events are undergoing, then the equilibrium again can be disrupted. Imagine again some kind of environmental factor or non-natural factor, whatever you want to imagine that impacts all of a sudden the normal course of life of a certain population. That is going to disrupt in equilibrium. All right, so this was a link where I would normally do click and simulate, for instance, what happens when the population size changes, especially that to show you how important is the population size to maintain this equilibrium. I'm not going to do the exercise right now for practical reasons, but I invite you to click on this one. It's a very easy link where you can actually set different population sizes and see what happens. Also, what you can do is also change the number of generations, and you can see how over the course of many hundreds of generations this equilibrium can be maintained constant. Allele frequencies can be maintained constant if you don't alter the population, and so on. So I invite you to click on this and kind of play on it. It really shows you nicely how this equilibrium model works. All right, so the Hardy-Weinberg equilibrium model serves as a foundation for understanding evolution. And because we can predict, we can understand where a population is going, what's going to happen to allele frequencies in certain situations, okay? And so far we talked about an equilibrium. So it's kind of an ideal situation, but it can be very realistic because all what we talked about, all those criteria, all those assumptions of the Hardy-Weinberg equilibrium models are many times, in fact, satisfied. But what will happen if there is no such equilibrium. Of some of these equilibrium assumptions are disrupted. For instance, what if a natural selection event comes into play? What if a disaster comes into a population? What's going to happen to this equilibrium? What's going to happen is known as genetic drift. So genetic drift is the change. So remember, equilibrium model states that allele frequencies will stay the same over the course of generations if all those assumptions are met. Genetic drift is the opposite, it's a change in the frequency of allele in a population. And there are many ways that this genetic drift can happen. Let's look at the main two types, of genetic drift that can happen in a minute here. And just to give you an idea, genetic drift, most of the time genetic drift can happen if the population size is very small. Here in this example, imagine that there are a few insects, population of insect where the number of insects is not very big on a sidewalk, and a person is walking there and happens to smash and kill some of these insects. Let's look at this initial population. You had a few individuals, and there was a little frequency of the green color. It was smaller than the brown color. Because of the size of the population and because of the randomness of the event that has happened, smashing just a person that walks on that sidewalk, you may end up with a population after the event that has quite different allele frequencies than the initial one. And if this population reproduces over the course of generations, you could even end up with the disappearance of one of the alleles that was present initially. So this phenomenon of change of allele frequencies over the course of generations is just an example of genetic drift. And in this example, the genetic drift is reducing the genetic variation quite a bit, okay, in a population. And so therefore the allele frequencies are going to be very different from the initial ones. All right, so this is the general concept, but as I told you, there are several examples of genetic drift. The two main examples we're going to talk about are the bottleneck effect and the founder effect. They're quite interesting to look at. Let's look at the bottleneck effect. The bottleneck effect refers to change in alleles or gene pool of populations to some drastic event that reduces the population size. Here is just a visual representation of what I'm telling you. For instance, imagine to compare this an initial or original population of a certain species. Take candies in a jar that has a bottleneck. You see that visually seems like maybe the blue and white alleles represented maybe 50-50, and then there is this yellow allele that is there, but is very at a percentage is much less than the blue and the white allele. Now imagine that for some reason you flip this bottle and you just take out of this bottle just a few candies. This could be a bottlenecking event, for instance, a catastrophic event, a fire or an avalanche that kills most of the population, in that place, and only a few individuals survive. And just by random chance, just like shown here, you'll have that the surviving population doesn't have the same allele fraction or frequency as the original one. Here I see that most of the survivors, just by random chance, were the blue, just very few, just one of the white, and none of the yellow. So if this surviving population starts reproducing, now you may end up with allele frequencies that are very different from the initial ones. So this is one example, bottleneck effect. And again, could be anything you can think about, even a parasite or an ecological event, a fire, something that disrupts most of individuals in a population, and just by random chance, the individuals that survive just because they were present in a specific place, maybe rather than another in a forest where maybe the fire happened, they happen to survive. Okay. Sometimes bottleneck effect can be caused by human. For instance, this is an example of northern elephant seals that were actually heavily hunted for blubber in the 1800s, and nowadays we have, because of this extensive hunting, we have reduced the number of individuals quite a bit, just because of random hunting. And the individuals that are actually that are alive, there are only a few colonies. They have very little genetic variation. So we have impacted the allele frequency, the genetic variation of these northern elephant seals because of this event. Okay, and again, it could have been any other, could have been just a change in temperature on Earth or things like that. All right, so just by curiosity, today there are just about above 100,000 individuals, again with very little genetic diversity. This was caused by hunting. It's again an example of bottleneck effect. All right, the other type of genetic drift that we can consider is known as founder effect. In this phenomenon, this phenomenon happens when a new population is established by a very small number of individuals, which are known as the founders. That's why this is known as founder effect. And by pure chance, the founder group maybe has a different allele mix or frequency than the original population. Again, you're extrapolating just few individuals from a population, and just by random chance could happen that population, the small population, doesn't really carry or is not representative of the same allele frequencies as the bigger population it comes from. There is a very good example actually of this founder effect in human population. There is an old order of Amish in Pennsylvania, which was established by only 200 German immigrants, okay, in Pennsylvania, and then they started, of course, this colony, if you will, if you want to call that. And remember, one thing here that is also important to mention is that Amish tend to marry each other and tend to marry also relatives in a small community. So not only do you have here just a few immigrants go elsewhere to found a new population, but you also have this kind of like disturbing element, which is disturbing compared to an equilibrium, of course, that is the really non the mating just within that population, not with the general outside human population where this small colony is established. And so I don't know if you are aware of, but this phenomenon in the old order Amish, there is a lot of polydactyly, which is a this is really a mutation. You can call it mutation here, but it's really an allele, a specific allele that determines 6 fingers and toes, and is very, very recurrent in the old order Amish. So what happened is that for some random occurrence amongst these 200 German immigrants, it just so happened that several of them carried this mutation, and because of this population being founded by this small number of individuals marrying each other over and over, now you have that there is a high incidence of this mutation or allele in old order Amish, which is not very representative of the incidence that you see in German population in general, or even in the Amish populations in Germany. So this is an example, a very good example of founder effect. Okay, simple as that. All right, so of course, as I told you, the interbreeding, because of the tradition of these people to marry people just in the same community, this has even increased the frequency of polydactyly. Okay. All right, so there is here a very nice video by the Amoeba sisters, which I'm gonna play for you, that explains genetic drift. Okay, so please watch this video carefully. It's really nicely made.
So here's a question for you. Have you ever compared the candy you get on Halloween with your friends? My sister and I, a bit competitive in the sport of Halloween candy hunting, we both had preferences of which kind of candy we hope to get in our buckets each year. And as for me, I love gummy bears. And every once in a while, someone would have them mixed in a big bucket, and I remember watching with intense focus the hand that picked out the candy hoping for gummy bears. Getting those gummy bears was always total luck, total chance. Well, unless of course the entire bucket they had was actually gummy bears, but that would not be luck. No, that would be pure bliss. But that's never happened to me yet. So why bring this up? Well, what if instead of candy, we were talking about insects? And they weren't in a Halloween bucket because that'd be kind of gross. They're on the sidewalk. And when you're scooter riding through the neighborhood on Halloween to collect candy because scooters may or may not be our super awesome childhood mode of transportation, you squash some. Accidentally, of course, because you like insects overall. Were the ones that survived better adapted? Did they have some special ability to foresee events or to move your scooter away from them? Well, assuming they were all just there on the sidewalk in the path of your trick-or-treating, then no. In this random event example, the survivors were not better adapted. They were lucky. It was chance. And the ones that were squashed were, in this case, at the wrong place at the wrong time. And that's what genetic drift is. Genetic drift can change allele frequencies in a population. Remember, alleles are a form of a gene, but genetic drift is random, unlike natural selection. Recall that in our natural selection video, organisms that have traits that result in high fitness are able to pass those alleles on to their offspring. This is where that survival of the fittest comes into play, but not genetic drift. With genetic drift, the organisms aren't necessarily more fit. They just have won the game of chance. So let's look at a representation of genetic drift known as the bottleneck effect. I always like to remember this one by focusing on the word bottle. Well, let's say that we have a bottle here and we fill it with our Halloween candy. If we shake it out and we only get a few of the candy pieces, does it necessarily represent the whole bottle as far as the candy frequencies you got out? Nope. In fact, one of the types of candy pieces isn't even there. That type is totally eliminated. We'll consider a natural disaster, such as a forest fire. The surviving organisms weren't better adapted. They were likely just in an area where they weren't directly affected. But the survivors don't happen to represent the original population, so there's definitely going to be a new allele frequency among the surviving population. Bottleneck effect. Another time to see genetic drift is when you're talking about the founder effect. In this case, you may have organisms that have founded an island or some new area. The few organisms that arrive to start a new population do not necessarily represent the original population that they came from. Let's take a case where maybe some seeds that are dispersed by wind end up in a new area that happened to be one that was ideal for their growth. This new population grows. Well, those seeds that landed in this new area may not necessarily represent the original population of plants from which they came from. It's a random sample, founder effect. In summary, genetic drift can make some big changes to allele frequencies, and it's a mechanism for evolution. So one last thing. Which type of population do you think would be more vulnerable to genetic drift? A big population or a small population? If someone is just doing some intense candy hunting and by accident scooter rides over a few insects, look at the results in the small population versus the big population. As you can see, this random event has the potential to cause more change from the original in the small population. See, there weren't as many representatives there in the first place. So in many incidences of genetic drift in populations, the impact on small populations is especially significant. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Okay. All right. So we learned about this phenomena. Okay. It's not that hard, I guess, to understand equilibrium and genetic drift. All right, so in the last few slides here of this presentation, I wanted to recall some concepts that we actually have studied already very well. The concept of natural selection and evolutionary fitness. If you remember, we stressed a lot the importance of this word fitness in evolution. Remember that in natural selection, the environment acts on population to select individuals with traits that are best suited for that particular environment, not necessarily the best, okay? And it's based on evolutionary fitness. Natural selection is based on evolutionary fitness in the sense that the individuals that can better survive and reproduce will pass on the traits that are more suitable for that particular environment. So remember, evolutionary fitness refers to a reproductive success and not to what we intend in the late terminology. So it's not necessarily the most fit individuals in a population in terms of, in evolutionary terms, are not the strongest necessarily, but are the best suited in the existing environment. Okay, sometimes traits that are not really good traits in general, generally speaking, could be actually favored and selected for in populations because maybe they do confer more evolutionary, reproductive success in that population. We talked about all this. And just a funny example. The example of sneaker males. This is seen in several species. This is a species of fish where there are two types of fish. There are some that are, in the males especially, some that are smaller, some that are bigger. This is the big fish. This is the small male. This is the big male. And the small male resembles a female. And I'm sorry, here it should say this is the female, I believe, and this is the male, but the female, the small male is very similar to the female. So there are two types of males in the same species. One is small and resembles the female. One is big and scary. So what happens here is that because the big male is not actually able to distinguish between the female and the small male, it allows the small male to stay around the female. And what happens is that while this female is depositing the eggs, the male will, the small male will be closer to the female and release the sperm, and so will have more chances of fertilizing the eggs than the big male. These examples are seen in several species, and this is known as the sneaker males. okay, where the Sneaker male is the one that actually fertilizes, not the biggest and strongest one, but the smaller one, okay? So remember, evolutionary fitness doesn't necessarily refer to the strongest or more fit individual, as we intend it in lay terminology, but the one that has more reproductive success, okay? All right, so we're done with this, and so again, in this presentation, we talked about all these points, very important points, which are your objectives, as well as the things that you need to know and understand from this lecture. Okay, thank you very much.